Okay. Okay, good morning to you all. The lecture today is biochemical basis of bone remodeling, and this lecture belongs to body tissues and locomotor system. And even though the time is given for 2 hours, but I will try to finish in 1.5 hours time. And some of the aspects may be discussed here in detail. I will emphasize what are the important areas you have to go through. Okay. So when you are first, we talk about the little bit recap about the bone structure. As you all know, the bone is a specialized connective tissue that, along with cartilage, form the skeletal system. And bone is not a static organ; it's a dynamic organ and highly vascularized and very active metabolically. Understand? As the bone are not completely developed at birth, they continue to be formed slowly out of cartilage or connective tissue. When the cartilage or connective tissue will get mineralized or calcified, the bone development takes place. So the cartilage and connective tissues are converted into the hard lamellar components of the bone. So what we trying to do today, we will introduce you the word modeling and remodeling. What is modeling? The growth of bones comes to an end at puberty with ossification of the growth plates. Understand? So the growth of bone from birth to adolescent, what happens? The modeling happens, right? The ossification, that means the bone formation in the growth plates, is known as modeling. About 90% of the adult bone is formed by the end of adolescence, and subsequent gains during adulthood are very small. So by the age of puberty, right, the bone formation or the ossification of the growth plates completes. There are two types of bone you would have done already. The thick, densely calcified bone that we know as cortical bone or compact bone, and thinner honeycomb network of calcified tissue that is known as trabecular bone. The compact cortical bone, the structure is given here, forms the outer layer of the long bones. Right? You can see that the outer layer of the long bones, that this is the, what you call that, the compact bone or cortical bone, we call it. And it's very densely packed and hard and has a slow metabolic rate also. Therefore, the cortical bone is resorbed and replaced at much slower rate, right, than trabecular bone. When you say resorbed, means the demineralization of the bone and then subsequently is replaced. When you come to the spongy cancellous trabecular bone, that is the one you can see when the cursor moves here, that is what you call the trabecular bone, right? Sometimes you know this is known as ossicles. The axial skeleton, basically the cranium, vertebral column, thorax, and pelvis has a specialized construction. Okay. So you can see the structure of the bone. This is the first one. Indicate the cortical bone. And then you can see the bone marrow and then the bone lamellae, right? That is expanded here, right? And then the osteocytes are there, the cells, right? This is called Cavesian system. And then this part of the bone is shown here, right? And then when you come to the trabecular bone, that is here, and then we divide this diaphysis, metaphysis, and epiphysis. And then the trabecular bone, this is the bone lamellae. And then this is the osteocytes. You can see the osteocytes. And that is what you call that bone marrow, right? This area is called bone marrow. Okay. And then when we go to the topic now, bone modeling, right? Modeling is differ from remodeling. The process of bone modeling is responsible for the formation and maintenance of the shape of the bone. So the initial stages from the birth to adolescence, your bone develops and it get a shape, right? And that shape is maintained and the structure is maintained, right? That process is known as bone modeling, right? So the bone modeling slowly happens throughout the birth to adolescent, and then at the end of the day leads to acquisition of peak bone mass. Right after that, as I said to you, the bone growth doesn't take place after adolescent or after you reach the puberty. Only slight changes take place after that. It begins in the fetal life, as I mentioned here, and continues until skeletal maturity, right? That is what we call that epiphyseal plate closure or completion of longitudinal growth of the skeleton. Okay. So bone modeling occurs. Removal of bone from one side and formation of bone at different side. That is what we call modeling. Remodeling is different from that. The remodeling is the removal of the bone and formation of the bone take place almost at the same place. But when it comes to bone modeling, removal of bone from one side and formation of bone at different side. And then the topic is now bone remodeling. We explain what is bone modeling. Now we talk about bone remodeling. The bone regeneration process persists even after skeletal maturity, right? Once the skeletal maturity means the modeling is over, right? The growth is complete. But sometimes we need to have the regeneration process has to take place after skeletal maturity. That means replacement of old bone with newly formed bone at the same location. That's what I said. Modeling take place, right? Removal of the bone is one place and formation of the bone is in another place. But here, right, the old bone is replaced by the new bone at the same location. That is called remodeling. So there's a difference between the modeling and remodeling. So whenever a bone is damaged, bone remodeling is required for repair of all damaged bone, right? Due to daily physical load and prevention of aging effects and its consequences. Understand? So remodeling take place, right? Any part of your life, right? You may have a fracture of a bone, right? That has to be repaired by remodeling, right? So when you have remodeling doesn't take place, if remodeling doesn't take place, right, that will always result in osteoporosis, right? There is a decline in the bone mass and density, right? So the impairment of the bone remodeling process often result in the progression to osteoporosis, right? So when you look at the structure of the bone, you would have done already. The main component of the bone matrix are collagen and hydroxyapatite. The collagen usually found in bone is usually type 1 collagen. Constitute almost 90% of all bone protein is type 1 collagen. And then what happened? The crystal containing hydroxyapatite, right? that Dr. Nisansala would have told you all what is hydroxyapatite, are found on top of the collagen fibers, in between the collagen fibers, right, within the collagen fibers, right? So the collagen fibers orientate so that they have a greatest density per unit volume, and it is packed in layers, right? When it packed in layers in these collagen fibers, the crystals are deposited. What crystals? Hydroxyapatite crystals are deposited such a manner the deposit can take place on, within, between the collagen fibers, right? And if that organic matrix is non-calcified, then you call it as osteoid, right? And then become mineralized through two mechanisms, right? Within the extracellular space of the bone, plasma membrane derived matrix vesicles. Plasma membrane form some matrix vesicles, right? Act as a focus for deposition of calcium phosphate that contain calcium phosphate, right? So that is the one, right? Leads to crystallization, right? The crystallization from the vesicles formed by the plasma membrane. After crystallization, right, the vesicle membrane, right, leaving the clustered hydroxyapatite crystals within the environment, the bone forming cells secrete the matrix proteins that rapidly mineralize and combine with matrix vesicle derived crystals. That is the way it happens, right? So the matrix proteins are secreted by the osteoblast, right? That rapidly mineralize and combine with matrix vesicle derived crystals. Okay. So when you look at this, there is pyrophosphate present in the matrix, inhibit this process. But the alkaline phosphatase secreted by the osteoblast destroys it, allowing mineralization to proceed. So you can see that if there is pyrophosphate means PP, right? If it is present in the matrix, that will inhibit what you call that, the crystallization or mineralization. That is why we have alkaline phosphatase. So that is one of the reasons we can see that we will be studying during the growth of the bone, alkaline phosphatase level increases. That is secreted by what? Osteoblast to destroy the pyrophosphate that usually inhibit the what you call mineralization of the bone. So allowing the mineralization to proceed, pyrophosphate is removed by the alkaline phosphatase. So the mineralization is highly dependent on the adequate supply of calcium and phosphate. So if there is a deprivation of the calcium or phosphate, result an increase in the non-mineralized osteoid. You know what I mean, right? Non-mineralized matrix of the bone is known as osteoid, right? For mineralization, we have to have adequate amount of calcium and phosphate. If calcium and phosphate levels are deprived or not adequate amount is available for mineralization, right? Then non-mineralized osteoid only you will get. That clinical condition is usually known as osteomalacia in adults and rickets in children, right? So you can see now here, there are two kinds. This is the blue color in this picture, right? The blue color is what you call that mineralized osteoid, right? Okay. Then you can see the osteoblast cells, right? In various shades of what do you call that? The osteoid is there. Mineralized bone is blue, and then the osteoid, the red color one, is actually right. Osteoid is going to be mineralized by the osteoblast, right? That's the structure of the bone, right? And you can see now, if the calcium and phosphate is not adequately available, right? If the osteoid, the non-mineralized osteoid is not mineralized properly, then what will happen? We will end up in a condition, right? The bone density decreases, and then that condition is known as rickets, right? This usually you can see it in the children, and if it happens in the adult, we call it osteomalacia. So, bone growth. Bone formation starts with the differentiation of mesenchymal cells into either chondroblast or osteoblast. So one is that means chondroblast or osteoblast. Chondroblast is the one is present in the cartilage, and osteoblast is the one present in the bone, right? Chondrocytes, these are the mature chondroblast, synthesize matrix protein containing type 2 collagen and proteoglycans forming cartilage. Chondrocytes subsequently enlarge, become the central cells of the growing bone, right? And they attract blood vessels and the chondroblast. The chondroblast is the one forming the, what do you call that, so-called osteoblast cells, right? So they usually digest the matrix, right? The chondrocytes subsequently undergo apoptosis, and the matrix they have synthesized is colonized by osteoblast, which commences bone formation. So initially, what happened? Chondrocytes do the function, and they undergoes apoptosis, and then what happened? That is colonized by the new cells that is called osteoblast, and they commence the bone formation. Eventually, the entire growth plate becomes bone and growth stops, right? So, all this bone formation and demineralization, right, under the control of several signaling pathways. The signaling pathways are important for the bone growth. The signaling pathway underlying chondrocyte maturation is known as Hitchcock pathway. Includes growing cartilage also secrete the parathyroid hormone related protein, right? So that is the parathyroid hormone related protein is involved in the Hitchcock pathway. Hitchcock pathway, parathyroid hormone related protein acts on receptors common with parathyroid hormone and maintains chondrocyte proliferation by regulating the Hitchcock pathway. This chondrocyte proliferation is also controlled by some growth factors, mainly the fibroblast growth factors. And there's another signaling pathway important in the development of the skeletal pathway, WNT, beta-catenin pathway, right? So the Hitchcock pathway and WNT beta-catenin pathway, both are important for the bone formation or skeletal development. Now we can see now in this picture, right? You can see the mineralized osteoid, right? That is, this is the bottom part, that is the bone, right? Mineralized osteoid. And then you can see in between, you can see the osteocyte, that is the bone maintaining cells, will come in a while, osteoid. And then this area, you can see there's a new bone formation. The bone forming cells are here, one layer, that is what we call osteoblast. Now these are the osteoblasts, right? And then you can see here, these cells are known as osteoblast. They are the one involving in what you call that demineralization, right? And then this part actually be known as what do you call that, bone marrow, right? So in that you can see the mast cells, hemopoietic cells, adipocytes, right? Bone marrow stromal cells, right? And blood vessels, so blood supply, highly supplied with blood, right? So this is osteoblast. At the same time, nearby you can have the osteoblast, right? So the interdependence of bone and bone marrow together, they form single structure functional entity, bone and marrow system. This is what we call the bone and marrow system. They are interdependent to each other. Now, when you look at a bone has two mechanical functions to fulfill, right? So when you look at this bone structure, right? The femoral head, neck, and shaft, right? The two principles of construction for a maximal weight bearing, right? The tubular structure illustrated by the television tower, that is the longitudinal one, and the trabecular structure by the crane, right? So you can see now the weight bearing action of this vertical structure, tubular structure, what do you call that, television tower like structure, and then the crane across, right, is resemble our bone structure. Femoral head and the, what do you call that, the shaft is equivalent to a crane attached to a, what do you call that, television tower. Okay, right. Now, this is a normal cortical and trabecular structure of a proximal femur. And then you can see what happens. When a patient with severe osteoporosis, the bone density decline, and then that look like this, right? So this is a bone which is having osteoporosis, and this is the normal proximal femur, right? Okay, now you can see that, as I mentioned you, right? Now we can see, suppose this is a femoral head here, right? When you have remodeling take place, usually I said, right, the demineralization as well as new bone formation take place at the same site, right? So you can see now, this is the lining cells. This is what you call quiescent state or resting stage. And then osteoblasts are recruited here. This is called resorption cavity, right? So here what happened? Demineralization is taking place. The old bone is removed by the osteoblast. And then what happened? Subsequently, osteoblast come into play. And then you can see the ash color. One is the new bone matrix formation. And then what happens? Subsequently, you get mineralization and the bone formed. Understand? So that what in roughly I wanted to say here, both bone demineralization as well as bone formation take place at the same place. The osteoblastic activity and the osteoblastic activity takes place at the same place, right? And as the body ages, the bone loses some of its strength and elasticity and therefore breaks more easily. This is due to loss of mineral and changes in the bone matrix. Bones undergo constant process for removal and replacement so that components of the bone are exchanged at regular intervals. That process is known as remodeling. So what are the purposes? Or otherwise, remodeling serves the following purposes. Mobilization of calcium in the framework of calcium homeostasis. That means the serum calcium level, right? When the calcium levels is low, what happened? Either the calcium can be contributed through the intestinal absorption of calcium from the diet, or else other source is what? Bone, right? So mobilization of the calcium from the framework for calcium homeostasis, right? Replacement of old osseous tissue, bone tissue. Then you need remodeling. Overall skeletal and individual local adaptation to different loads, weight bearing and stress. So local adaptation, right? So you need the remodeling, right? For that, you can have what you call that, bone remodeling. Sometimes a bone can get damaged. To repair a damaged bone also, remodeling helps, right? So these are the purposes of remodeling. So we go into detail of bone cells. What are the type of bone cells we have? One is we call osteocytes. These are involved in maintenance of bone tissue. Then osteoblast forms bone matrix. Then osteogenic cell, they are the stem cells. And then osteoblast resorbs bone. So one is bone resorption, other one is bone formation, other one is maintenance of the bone tissue. Maintenance of the bone tissue is done by osteocyte. Formation of the bone matrix is done by osteoblast. And then resorption of the bone is done by osteoblast. And the stem cells from that, these osteoblast and osteoblasts are derived, is known as osteogenic cell. So there are 4 types of bone cells. So when you look at this, osteoblast and osteoblast are evolved from stem cells, right? There are 2 types of stem cells. One is called HSC, hematopoietic stem cells, right? They differentiate into mononuclear cells, and they differentiate into, they fuse together and then form polykaryon. The polykaryon is differentiated into osteoblast, right? And you can see this is where a bone structure, right? The osteocyte is mentioned, shown in yellow color. That is involved in the maintenance of the bone, right? And then this is the old bone. And now this is the new bone. The black and below that is called old bone. And now this is the orange color one is a new bone. And then the yellow color one is what you call that bone matrix, not mineralized yet. Okay, that is osteoid. And then you can see the bone forming cells are known as osteoids, right? And then they are derived from another type of stem cells that is called mesenchymal stem cells, MSC. And then they differentiate into osteoblast progenitor, pre-osteoblast, and the pre-osteoblast become osteoblast, right? And then you can see these yellow color cells are known as bone lining cells, right? So this is how, right, evolution of osteoblast and osteoclast take place from the hematopoietic stem cells as well as mesenchymal stem cells. So we'll go into detail one by one each bone cells, right? What we are going to discuss is about osteoblast, osteogenic cells we have already seen in this diagram, right? So we will go into detail of osteoblast, and osteocyte. First, we take what is osteoblast. The osteoblast, another name for osteoblast are bone resorbers, right? They resorb the bone, or bone breakers, right? Okay, all bone cavers can resorb old weak bone in a short period of time. You can see now they are multinucleated cells. So this is this is an osteoblast, right? This is a multinucleated giant cells. The cells are large in size. Why they have become multinuclear, you can see now, right? In this diagram, hematopoietic stem cells, right, differentiate into mononuclear cells, and then they fuse together, and then what happens? All the nucleus of these individual cells come together. That's why we call fused polykaryon, right? So you can see that they are multinucleated giant cells, and they are derived from monocytes. What is monocyte? And then this is what you call monocytes. Okay, right. And form from HSC, lifespan up to 7 weeks. They have the lifespan 7 weeks. The cell membrane consists of numeral folds. Folds, right? This is the numeral folds. This is called ruffled border, right? The fold are called ruffled border. So that is the characteristic of osteoblast, right? Multinucleated, derived from monocytes of the bone marrow, and that is from hematopoietic cell line, right? Lifespan is 7 weeks, and the cell membrane consists of numeral folds. That's called ruffled border. So now looking that here, you can see that, right? Now this is the bone. Okay, you can see this is. I'm sorry, the cursor isn't coming. Okay. All right, here. Now this is the bone, right? Okay. Now here, the remodeling is going to take place here. That means this part of the bone, right? You have going to have the resorption and subsequently the bone formation. Right? Okay, right. Now, very carefully listen to me. We have mentioned something here. What is that? In this picture. Right? Now, this is called, in this picture, you can see this cavity. First, there's a cavity formation will take place. That cavity is known as resorption cavity. Now, this is individual, what do you call that? Osteoblast, these blue color ones, right? So, now this is the resorption cavity. This is a resorption cavity. Okay. So, you can see now, I have shown you one osteoblast cell that is attached to this bone by integrin. The blue color one is called integrin. Right? So what is the major function of the osteoblast? Right? That is to destroy the surface of the old bone, old and fractured bone. Right? And then allow the osteoblast to come in to form the new bone. So what is actually needed in the bone tissue? You have matrix as well as protein. Understand? So you need what you call that proteoglycans, right? As well as, right? You have what you call that collagen. So you need what have a lot of proteolytic enzyme and other enzyme is essential for the demineralization of that area. Right? Okay. So that is an extracellular matrix. So these proteolytic enzymes are matrix metalloproteinases. Right? By the action of matrix metalloproteinases, what happened? These bones get dissolved. So how it has happened, Kilabalamopi? Right? Now, here you can have a transporter, chloride ion comes in, HCO3 minus goes out. Potassium comes in, sodium is goes out. Understand? Right. Because why chloride, and here you can see now, chloride ion is coming here. Chloride is coming here, right? Transported to this cavity. And then you can see H plus is transported to this cavity. Right? When the chloride ion transported, you need more chloride ion inside the cell. That is by another mechanism that is called HCO3 minus goes out and chloride ion comes in. And then you can see potassium, right? This is H plus ATPase. And then so this H plus and then Cl minus form an acidic environment. That acidic environment dissolve, what do you call that? The bone material. Not only that, you can see now, this purple color one is a lysosome. The lysosome also secrete enzyme. These enzymes are called lysosome enzyme. So the lysosomal enzyme also coming here, right? So what happened? Demineralization, the bones get dissolved, right? So all these enzyme, this acid, lysosomal enzyme, and then proteinases, right? Now, and then phosphatases. Now, this is TRAP, you can note it all. Tartarate resistant alkaline phosphatase. Tartarate resistant alkaline phosphatase. So that means that is again involving in the dissolving of the bone. Understand? So bone particle, the fragment of the bone will come when that all the acidity, enzymes, lysosomal enzyme, proteinases, right? Most of the time the proteinases are matrix metalloproteinases. That is involving the remodeling of the extracellular matrix. The enzyme is known as matrix metalloproteinases. All these dissolve the bone here. They are in this resorption cavity. And this entire process, now you can see once, sometimes you get the fragment of bone will be there, right? So we have to remove that. Before osteoblasts come in and form the new bone, the fragments has to be removed. So that is what happened by this osteoblastic cell, by a mechanism called phagocytosis, take up the fragment of the bones and then inside and destroy them. Understand? So you can see now, whatever the calcium, phosphate, and peptides, right, taken in vesicles from this area and then removed by exocytosis. Removed by exocytosis. So you can see now, there are various factors, right, involving the resorption. Some of the factors resorption are stimulated by the factors. Bone resorption stimulated by the factors. And some factors inhibit the bone resorption. As if you look at this way, right, parathyroid hormone, right, stimulate bone resorption. Interleukin 1, IL-1, stimulate bone resorption. TNF, right? Okay, that is tumor nuclear factor, stimulate. TGF alpha, it's a growth factor, tumor growth factor alpha, stimulate bone resorption. LH, stimulate bone resorption. Understand? Some of them inhibit the bone resorption. You can see that estrogen, calcitonin, beta interferon, TGF beta. You can see TGF alpha stimulate the bone resorption, TGF beta inhibit the bone resorption, right? So there is a signal transduction mechanism is involved. For example, calcitonin, how it inhibits the bone resorption. If you ask me a question, right? Calcitonin will, you can see that we will study later on. When you compare to PTH and calcitonin, they act opposite to each other. PTH is parathyroid hormone secreted by the parathyroid glands. Calcitonin is secreted by the parafollicular cells of the thyroid gland, right? Follicular cells secrete the thyroid hormone, parafollicular cells secrete the calcitonin. So PTH stimulate the resorption, bone resorption, and calcitonin inhibit the bone resorption. How the calcitonin act? The calcitonin, you can see that the purple color one, it will bind to its receptor. And ATP is converted to what you call the cyclic AMP. You call cyclic AMP. This is called pyrophosphate. Can you remember how the pyrophosphate is, when the pyrophosphate is present, what happens? I told you, bone resorption will take place. Can you remember? Right? When the bone formation take place, what happened? Bone growth take place, what happened? This pyrophosphate has to be removed. How we are going to remove the pyrophosphate? right? This pyrophosphate will be removed by alkaline phosphatase enzyme. That is why during the bone growth, pyrophosphate is removed. That is why we call alkaline phosphate elevated in bone growth. Not only in bone growth, but also in the, what you call that, wherever we have the high osteoblastic activity. Okay. So now this is how osteoblast cells. Now this is the osteoblast cell, multinuclear cells you can see, right? The trap here, the trap means tartarate resistant alkaline phosphatase, right? Proteinases, chloride, H plus come together, and the environment will be acidic, so that bone will dissolve, and the lysosomal enzyme will digest the proteoglycans, collagen, etc., etc. Proteinases also digest the proteins, right? Okay, and then phosphatase enzyme, everything happens here, and finally, mineralization, resorption of the bone take place. Understand? So this is an action that this is what you call that. Now this is the bone. Now this is the ruffled border, and then this is what you call that osteoblast, right? Okay. So the major function of osteoblast to destroy the surface of the old and the fractured bone. So the osteoblast releases of quantities of proteolytic enzyme and other enzyme into the space between the ruffled membrane and the bone, right? Between the ruffled membrane. So now this is the ruffled membrane and this is the bone. So that's the cavity. That cavity is known as resorption cavity. Okay. Right. Now, when you come to this, we have to understand now very carefully. The activation of osteoblast, right, under the control of hormones, right? That is called systemic hormones. This part is very difficult to understand, but we have to understand also. Systemic hormones, such as parathyroid hormone, estrogen, androgen, leptin, thyroid hormones, as well as cytokines, and various growth factors also involved. And then recent investigation came across a pathway, rank and rank-KL signaling pathway, right? The rank and rank-KL signaling pathway in the osteoblast have clarified mechanism of stimulation of activation of resorption. How the resorption take place, how this activation take place, right? Explained by a pathway known as rank-KL signaling pathway. Understand? So, rank is the receptor, right? The rank-KL is a signaling molecule, right? So we go to the next slide, you can see that, right? So that receptor rank consists of, what do you call that? The structure, you can see that. Osteoblast possesses a membrane receptor structurally related to tumor necrosis factor receptor. The receptor is named as receptor activator of nuclear factor, NF-kappa B, right? The simple case called kappa. Receptor activator of nuclear factor, NF-KB, that is called rank, right? So now this is the rank, right? This is rank. Okay. This is rank. And its ligand is TNF related cytokine called, right, rank KL. So that means rank ligand. It's a cytokine, right? The ligand is T. We don't give a name for the cytokine, and then rank is the receptor, but the molecule comes and binds to the rank is called rank ligand. So it's called rank KL, right? This is the receptor is known as rank the molecule, the ligand comes and binds to the receptor. Now this is rankale. Now this is rank. Now this is the receptor. Receptor to bind when rankale. Okay. Rank signaling involves recruitment of an adapter molecule known as TNF receptor associated cytoplasmic factor. So you can see what happened. There is an adapter molecule. I want to show you adapter molecule. Now this is what you call that TRAF. TRAF, this one, right? TNF receptor associated cytoplasmic factor. That will come and bind to the, now this is what you call that, right? Extracellular domain, then the transmembrane domain, and then the intracellular domain, right? Right? Understand? So what happened? It will come and bind to the RANG molecule, TRAF, bind to the RANG. When it binds to the RANG, there are subsequent signal transduction pathways will be activated. That is, I show you here. So when it binds to that RANG, right, RANKL will come and bind to the receptor. So what happens? There is a signal. So the different kinases will be get activated, right? So many pathways will happen. I'm not going to detail of those pathways, but something you should know, right? So what happened? There is a protein will get activated that is called SRC protein, and then this is PI3K kinase, AKT kinase, MTOR kinase, like that is get activated. So when these kinases are get activated, you can see all these are kinases, K, they end up in K. What happened? Some of the transcription factors will be activated. Understand? So when the transcription factors get activated, what happened? The particular protein in the DNA will get activated. That will lead to what you call the bond resorption. Understand? So the kinase activation in turn leads to activation of array of transcription factors, right? That is what you call NFKB, nuclear factor, kappa B, and AP1. All these induction of genes coding for what? Tartrate resistant acid phosphatase, cathepsin, calcitonin, and beta-2 integrin. What they will do? You can see beta interferon, calcitonin, right? Cathepsin, right? You can see. Now this is what you call that tartrate resistant acid phosphatase. That is phosphate part of the bone is dissolved by that enzyme. Understand? So various enzymes related to what do you call that bone resorption will be stimulated by this pathway, right? Okay. So the kinase, again I am coming here. RANKL is the ligand for the RANK receptor. RANK receptor is attached to a cytoplasmic component that is called TRAF. When they are attached, this receptor will be active and then allow the RANKL to come and bind. And that will activate a signal transduction pathways where some of the kinases will be activated. That kinases activation lead to what you call that some transcription factors production. These transcription factors bind to the DNA. And then these genes coding for what you call the tartrate resistant acid phosphatase, cathepsin, calcitonin, beta-2 integrin, they control the bone resorption. Understand? So the RANKL signaling pathway is important for the resorption to take place. Right? So osteoblasts are controlled by local factors and systemic hormones. So some of the factors are local factors, some of the factors are systemic hormones, right? So this is always what happened. Cytokines such as interleukin, tumor necrosis factor, transforming growth factor, all I shown you, right? Transforming growth factor beta, interferon alpha, control the osteoblast actin through RANKL. And there is another term I am going to introduce, right? OPG. OPG means osteoprotegreen, or protegerino, OPG, a decoy receptor, right? RANKL can bind to the OPG. When the RANKL bind to OPG, is a receptor. It's called decoy receptor. Decoy receptor, right? Resembling the structure of rank. So the RANKL can bind to the OPG also. You can see now. This is OPG. This OPG, RANKL goes and bind here means the signaling pathway won't take place. Then the resorption won't take place, right? So next diagram is important. Very carefully listen me. Okay. Now this is the bone. Okay. Everybody understand what is this? This is a bone. Right. Now this is what you call bone resorption cavity, isn't it? Now this is the bone resorption cavity, isn't it? Right. Now what is this? The yellow color one is what? Osteoblast, isn't it? Yellow color one is osteoblast. So you can see chlorodyne secreted into this. When the chlorodyne secreted into this, what happened? Chlorodyne has to come to the osteoblast. Bicarbonate ion, chloride anion exchange HARA, chlorodyne is coming in. And then you can see carbon dioxide and H2O in the osteoblast. By the carbonic anhydrase enzyme, it formed the H2CO3. H2CO3 ionizes and produces H plus via ATPase, H plus ATPase enzyme, H plus comes inside. And then the pH will become acidic now. Why pH become acidic? Because of the HCl. And osteoblast has a mitochondrial activity. So the mitochondria of the osteoblast will produce citric acid. Citric acid also transported via the citric acid transporter. So that also give you the resorption pit and acidic environment. Acidic environment. Right? Now, because of this acidic environment, because of these lysosome enzymes, right? Okay, this is a ruffled border. The bone will get dissolved. Collagen will be digested. Right? From the matrix, calcium and phosphate will be, but it's a hydroxyapatite. Calcium and phosphate is there. Right? That will be undergo dissolution. And then you can see now the collagen fragments are removed by endocytosis, right? By vesicles from the pit, resorption pit. This goes here by the endocytosis. The collagen fragments are removed, and then calcium and phosphate are removed, and the bone is dissolved now. Bone is dissolved. Who is stimulating this? This fellow has. How osteoblast will have rank. Rank eකට බයින් වෙන්න කවුද? RANKL. RANKL is coming from whom? Osteoblast. Very interesting, isn't it? The receptor for RANKL is located in the osteoblast. Understand? But the RANKL is produced by what? Osteoblast. So when the RANKL comes and binds to the rank, very careful, RANKL come and bind to the rank, the entire process will be stimulated, bone resorption will take place. Bone resorption will take place. Now, I told you there is one called OPG, isn't it? I told you OPG. Now this is rank KL rank bind. So rank KL bind to rank, right? Rank located in the osteoblast. Rank KL is produced by osteoblast, right? There's one called OPG. What is OPG? Osteoprotein, OPG. What is that? It's a decoy receptor. So the decoy receptor, right, can go and bind to this RAN-KL. So when the decoy receptor, so that means RAN-KL can bind to this, or RAN-KL can bind to OPG. If the RAN-KL bind to rank, bone resorption will take place. That is called stimulation of the bone resorption. If you don't want the bone resorption, if you want to stop the bone resorption, what happened? The RAN-KL will go and bind to the OPG. When it binds to the OPG, now this rank will be empty. So that doesn't stimulate the bone resorption. That's called only inhibition. So that is what you call that. When the bone remodeling take place, first what happened? Osteoblasts come into this bone where you need the remodeling, and then the bone resorption will take place. That means dissolution of the bone. Old bone is removed. Understand? Then what happened? Osteoblasts come to that area. This osteoblast will be removed by apoptosis. And then what happened? Osteoblast will come and start forming the bone. Right? So now we studied about osteoblast now. Now we are going to talk about what? Bone builders. Bone builders are what? Osteoblast. Right? So they are derived from mesenchyme, isn't it? And you can see this. Hematopoietic stem cells produces osteoblast. So osteoblast is derived from hematopoietic stem cells. MSC, mesenchymal stem cells, leads to differentiate and then produces osteoblast. So osteoblast is bone builders or bone forming cells, right? They derive from mesenchyme of the bone marrow. They are cuboid cells that form the continuous layer usually in the bone surface. The lifespan of osteoblasts range from 1 month to about a year. They produce a new bone of several weeks to replace that resorbed osteoblast. So osteoblast does the resorption. In that place, osteoblast will come and form the bone. So the main function is what happened? Bone matrix, especially collagen type 1, and also osteocalcin, osteonectin. These are some of the protein, morphogenetic proteins. And then osteoblast also possess a receptor for estrogen. We'll come to that. So now you can see now, after the osteoblast activity, now this layer, you can, the blue color layer is what you call that osteoblast. It forms a continual epithelial surface. Now this will start at producing the new bone. Now you can see this second diagram. The new bone formation will take place by the osteoblast. Understand? So the bone builders are osteoblast. And then once the bone is formed, then we have to go to the bone cell osteocytes. They are bone maintainers or bone controllers, right? So you can see that we have done already. There are 4 types of cells. Osteogenic cell is the stem cell, that is hematopoietic stem cells, as well as, right, mesenchymal stem cells. That is hematopoietic one is HSC, forming the osteoblast. Mesenchymal stem cell forming the osteogenic cell. These are the osteogenic cells, I am sorry, stem cell. And then the osteocyte, osteoblast, and osteoblast. So we have done the osteoblast in detail, and we have done the osteoblast in detail, and we are going to talk about the osteocyte. Osteocyte is what? Bone maintenance, we call it, right? Bone maintenance and bone controllers, most numerous of the all bone cells, they develop from osteoblast. Approximately every 10th osteoblast is situated on the surface of the bone, is entrapped inside the bone matrix and become osteocyte. Right? Osteocyte lose their ability to secrete bone matrix. Osteoblast can secrete the bone matrix, osteocyte cannot. Very careful, right? Osteoblast can secrete the bone matrix, but osteocyte cannot. right? So osteocytes occupy the space in the bone called lagunae, right? And are connected to each other endostele cells on the surface of the bone by thin channels called canaliculi. I'll show you in a diagram. So you can see this. Now, this is what you call that, the bone, formed bone. Now, this is the Now, this is the surface, isn't it? In the surface, you have the thin layer of what you call, this is osteoblast. Now, this is the new bone, right? Now, this is the central canal. You have the nerve as well as blood vessel, artery, and the vein. And then now this is what you call lamellae, right? Okay. In between the lamellae, you can have this. These are the osteocytes. This is osteocytes. And they are connected by small canals like that. That canal is called canaliculi. Canal is called canaliculi. So you can see that this is here in this diagram also, you can see that after staining, this is what you call that osteocytes. So from osteoblast, one in every 10 osteoblasts is drawn into the bone formed or bone structure and then become osteocyte. That is for the maintenance of the bone. They don't have the ability to secrete the matrix just to maintain the bone. So you can see that. Now this is the red color one is osteoblast and the blue color one is osteoblast and then osteocytes. So these are the different type of bone cells. And then we are going to now bone remodeling units. In a bone, there are 2 to 5 million bone remodeling units are there, right? In the skeletal system, right? So when you look at a bone remodeling unit, BRU, what are the cells you get in the bone remodeling unit? Bone resorbing cells, osteoblast, bone matrix synthesizing cells, osteoblast, and bone protecting cell is called osteocytes. Understand? And progenitor cells. We are not talking about the progenitor cell. Understand? So look like the bone remodeling unit, right, required for the maintenance and integrity of the bones are of crucial importance for the development of osteoporosis. The total quantity of bone decreases if more bone is resorbed. That is what I told you, right? If the resorption increases, the total quantity of the bone mass decreases over the years. It has been estimated osteoporosis develops when every 30 units of bone resorbed, 30 BRU, right? Different units have. Every 30 units of bone resorbed, only 29 are produced. Then what happened? Osteoporosis developed. For example, that is called negative bone balance. The negative bone balance leads to osteoporosis. What is the reason for the negative bone balance? Increased osteoblastic activity without increased osteoblastic activity. 30 osteoblastic activity tibunot bone, what do you call the resorption? Bone formation happening only 29 mean that is again bone balance is negative. So increased osteoblastic activity without increased osteoblastic activity, that's called high turnover, right? Normal osteoblastic activity but decreased osteoblastic activity. Osteoblastic activity very well ne. Osteoblastic activity decreased. The low turnover. Decreased osteoblastic and osteoblastic activity, both. That is an atrophic condition or adynamic bone. We call it adynamic bone. If the both activities at high level is the dynamic bone, bone is a tissue, is a dynamic tissue. But both osteoblastic activity tadwela, osteoblastic activity tadwela tibunat atrophic or adynamic bone. So there should be a balance between what you call that osteoblastic activity and osteoblastic activity. Osteoblastic activity variable that will lead to osteoporosis because bone mass will decline. So when you come to this bone resorption unit, there are one remodeling take place. Bone resorption, subsequently bone formation and mineralization. It will take 120 days. So it can be divided into 6 phases. Phase of quiescence, phase of activation, phase of resorption, phase of reversal, phase of formation, phase of quiescence again. So that is very nicely you can see. This is what you call a BRU. BRU means bone resorption remodeling unit. I'm sorry. Beautiful, isn't it? You can see that the osteocytes are in the old bone and the new bone also. You can see the osteocyte. Osteocytes are derived from what? Basically osteoblast. Osteoblast goes inside and become osteocyte. When they are in the osteoblastic state, they can form the matrix. Once they become osteocyte, they cannot form the. Now, these are bone lining cells, right? Bone lining cells. And then you can see osteoblast, the giant cell recruited, right? This is the border, right? Here, this is a multinuclear cell. Then you have the macrophages here, then osteoblast. And now this is the old bone. Now this is the new bone. And then this is called osteoid. Osteoid is non-mineralized bone. Non-mineralized bone. Now you can see there are 6 phases I said. This is the quiescence where the nothing happening. Bone is covered by the bone lining cells. The second stage, we go resorption. Osteoblastic activity starting, right? Bone resorption take place. I already explained the mechanism of rank and Rankale. Understand? Then what happened? Reversal, right? Reversal period is where the osteoblast is removed from the area. And then what happened? Formation. Formation is what? Osteoblast activity. New bone is formed. After that, what happened? Mineralization take place. The osteoblast activity result in osteoid, non-mineralized bone tissue. Then mineralization, and then the new bone is formed, and quiescence. So there are 6 phases of remodeling. This is BRU unit. BRU unit means where the bone remodeling taking place. That is called bone remodeling unit. Right? Phase of quiescence. A layer of flat lining cell over the thin collagenous membrane covers the surface of the bone. Nothing happening. Then we come to the phase of activation. Right? Phase of activation, that is the surface is prepared for resorption. Right? Endosteal lining cell, removal of the thin collagenous membrane is removed now. Right? Matrix metalloproteinases produced by the osteoblast are involved in this process. Collagen membrane is removed, covering the surface of the, that means the membrane, the layer of flat lining cells. The enzyme is matrix metalloproteinases. Understand? Then the phase of resorption, osteoblast will come in, started the resorption, right? Then the phase of reversal. Osteoblast progenitors are attracted to the resorption pit, while monocytes and endothelial lining cells prepare the surface resorption pit for new bone production by removal of the debris by the osteoblast. So the debris will be removed by the monocytes and the endothelial lining cells. I can show you in the picture, then it's easy to understand. Right? Okay. So this is the bone lining cells, osteocyte. You can see the osteoblast activity. After osteoblast activity, macrophages and monocytes come into play, and then they remove the debris. And then you can see this is coming in. What do you call that? Osteoblast come in, and the new bone formation take place. Right? The quiescence, resorption, reversal, formation, mineralization, and quiescence again. Understand? So this is what you call bone remodeling. Okay, so the bone remodeling take place in various places, right? Now, some of the factors usually, what you call that, inhibit the osteoblast activity. Some of the factors, right, stimulate the bone formation activity, right? That is how the regulation take place, right? I'm not going into detail of that. The systemic growth factor and hormone involvement in this. What you should know is what are the factors stimulate, that is in this picture. You can see that. What are the factors inhibiting the osteoblastic activity? What are the factors stimulating the osteoblastic activity? If you know, that is more than enough. Understand? So what are the factors stimulate the osteoblastic activity? What are the factors inhibit the osteoblastic activity? Is more than enough, right? And then that detail will complicate everything. And then, but you should know something about the BRU, bone remodeling unit, and what is happening on the BRU, you should know. Other than that, this detail, if anybody can read and understand this, fine. I welcome you, right? Other than that, it's nothing much in that. Okay. So then what happened? The calcium regulation, calcium homeostasis, right? That will be done by physiology department, right? About the action of calcitonin, parathyroid hormone, and vitamin 1, 2, 5, dihydroxyhormone and estrogen involvement. That is not belongs to biochemistry, but I have included in my note also. Once you finish the physiology lectures, you can go through this. Understand? Then I have given you some of the biochemical markers for bone remodeling. Some of the markers are bone formation markers. Some of the markers are bone resorption markers. Just to have an idea, what are bone formation markers, what are bone resorption markers, just to have an idea, it's more than enough. Okay, so at the end of the day, what we have done, what is bone modeling, what is bone remodeling, and then structure of the bone overview, we have done it. And then bone cells we have done. What are the bone cells? There are 4 types of bone cells. One is osteoblast, and osteocytes, and then progenitor cells, etc. And then we have talked about how these osteoblast cells are derived, and how the osteoblast cells are derived from their stem cells. And then we have talked about the resorption pit. What is happening in the resorption pit, right? The mechanism of resorption, the involvement of rank and rank-AL, and the signal transduction pathway. When the resorption is over, how this osteoblast is recruited and the new bone formation is taking place, that is called osteoid. And subsequently, what happened? Osteoid will be mineralized by the calcium phosphate, and that is become new bone. And then what happened is osteoblast, 1 in 10, right? Osteoblast will be taken inside the bone, become osteocytes, and they are involved in the bone maintenance of the bone. And then what happened? There are growth factors and systemic hormones involving in the regulation of what you call that bone resorption and bone formation, right? Some of the factors I listed there in the diagram. If you know what are the factors inhibiting osteoblastic activity, what are the factors stimulating osteoblastic activity, what are the factors stimulating the bone formation, if you know that, it's more than enough. So that is all about biochemical basis of bone remodeling. Do you have any question from this? Have you all followed very well? Understood very well? Any response? Hello? Hello? Hello?
Yes, sir.
Okay. Anything you want to ask now? Or you want to ask later in the tutorial? Okay, you read and come. We have a tutorial, and then most probably the tutorial will be on Wednesday. And we'll do some question, and then you will understand more. The one chat is there. What is that chat is? So could you please send the recording? Yeah, it can be given. Don't worry. Okay, now I will upload the tutorial questions now. And then so you go through the tutorial question and then read the notes and then come back. Do you want a textbook? to share with you? Hello? 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 148 students are there. Do you all want a textbook for this to read?
Yes, sir.
Okay, right. So I'll wind up here and then we'll meet you again in the tutorial. Okay. Thank you. Take care. See you in the tutorial. Bye.